Welcome to another deep dive.
Yeah.
Today we're embarking on an ambitious exploration into one of the most fundamental and complex puzzles of biology, the intricate mechanisms of genomic instability and telomere attrition. This isn't just an abstract scientific concept. It's the very bedrock upon which the process of aging is built, a topic that spans the breadth of genetics, molecular biology,
Yeah, absolutely.
and even some truly surprising medical interventions that are emerging from this foundational understanding.
Indeed. And we've meticulously compiled a rich tapestry of information for you, drawing from seminal research, compelling real-world case studies, and the very forefront of cutting-edge discoveries.
Okay.
Our objective for this deep dive is to provide you with an exhaustive and comprehensive understanding of precisely how our DNA, the very blueprint of life, and the specialized protective caps on the ends of our chromosomes, known as telomeres, collectively dictate the pace and trajectory of cellular aging within our bodies. Our mission is not simply to describe, it's to meticulously unpack the complex molecular mechanisms at play.
Get into the details.
Exactly. To illuminate their critical consequences for human health, and to cautiously explore the exciting, albeit carefully considered, avenues for therapeutic intervention that are being developed.
Think of this as an expedition, a systematic peeling back of the layers on one of life's most persistent and universal mysteries. Why do we age? What are the intrinsic biological forces driving this inevitable process? And perhaps more importantly, what can we, as intelligent and curious observers, truly understand and potentially even influence about this complex biological phenomenon?
Right.
To embark on this journey, we'll begin with a powerful real-world scenario that starkly highlights the immense stakes and profound consequences of genomic damage.
Yeah, a really grounding example.
From there, we'll plunge into the intricate molecular nitty-gritty of how our cells manage DNA repair, the fascinating biology of telomeres, and the cascading ripple effects that these molecular events have throughout the entire human body.
It's all interconnected.
We'll then pivot to examine some truly groundbreaking research and the novel therapeutic approaches that are emerging from these insights. Before concluding, we'll delve into practical measures you can actually take to protect your genomic integrity and, critically, how scientists are able to precisely measure and track all of these molecular changes.
Very important, the measurement aspect.
Finally, we'll grapple with the critical balance of risks and rewards that accompany our growing ability to manipulate these fundamental processes as we gaze into the future of anti-aging interventions.
Ultimately, this deep dive is designed to equip you with a robust, scientifically rigorous framework for understanding the core biological drivers of aging. It aims to offer insights that are not only deeply rooted in molecular science, but also profoundly relevant to our pursuit of enhanced health and extended longevity. So let's begin our meticulous unpackaging of this vital subject.
To truly set the stage for our deep dive, let's begin with a stark, tangible, and profoundly impactful real-world illustration of genomic instability in action. We're going to look at the silent yet devastating consequences of radiation exposure, a clear environmental challenge to our genetic integrity.
A compelling and indeed somber example comes directly from meticulous studies of workers who have been occupationally exposed to ionizing radiation over prolonged periods.
Like in nuclear facilities or medical imaging.
Exactly, places like that. These groups are often meticulously compared to carefully matched control groups who have had no such exposure.
Right, the controls are key.
Absolutely. We know with scientific certainty that ionizing radiation, even at relatively low levels accumulated over time, directly inflicts damage on the very structure of our DNA.
It's not just theoretical then.
No, not at all. This damage isn't merely theoretical or abstract. It manifests as clearly observable and quantifiable changes at the chromosomal level within these individuals, providing us with direct, undeniable evidence of its profound biological cost.
So how precisely do scientists observe this damage? What are the specific telltale signs they look for within the cells of these exposed individuals that indicate their genetic material has been compromised?
Researchers can, with remarkable precision, detect higher frequencies of specific chromosomal abnormalities directly within the blood cells of these individuals, particularly their lymphocytes.
Okay.
For instance, numerous studies have consistently shown that radiation workers exhibit significantly elevated levels of what are scientifically termed chromosome aberrations.
Aberrations. like mistakes.
Kind of. To visualize this, imagine your chromosomes, which are typically highly organized, thread-like structures made of DNA tightly wound around proteins, perfectly aligned and meticulously intact within the cell nucleus. In the context of aberrations, you might suddenly see these ordered structures showing breaks, like a physical rupture in the thread. Or fusions, where two chromosomes or fragments incorrectly joined together, or even complex rearrangements where large segments of DNA have been misplaced or inverted. These alterations from the normal, ordered, and stable chromosomal architecture are direct, quantifiable evidence of severe and impactful damage.
So we're talking about tangible, physical changes to the chromosomes themselves. It's almost like seeing structural cracks or breaks appear in the foundational beams of a building.
Exactly. That's a very apt analogy. It's visible evidence of structural compromise.
Okay.
And another crucial marker that scientists meticulously track is the presence of micronuclei.
Micronuclei. Tiny nuclei.
Pretty much. Picture a normal, healthy cell nucleus, which is a perfectly organized central compartment containing all your precious genetic material neatly bundled into chromosomes.
Right.
Micronuclei are distinct, tiny, separate nuclei, or more precisely, small extranuclear chromatin bodies that have literally broken off from the main nucleus.
How do they form?
They form from chromosome fragments that were either directly broken off during a damaging event, like radiation exposure, or from entire chromosomes that were improperly handled during the incredibly precise and intricate process of cell division.
During mitosis.
Exactly. And thus weren't correctly incorporated back into the main nucleus of the daughter cell. Both chromosome aberrations and micronuclei are direct, tangible, and measurable indicators of genomic instability.
So they tell us something's wrong.
Unequivocally. They tell us that the cell's genetic material is not only being damaged, but is also being processed, partitioned, or repaired incorrectly.
And these microscopic markers aren't just fascinating academic curiosities for researchers, are they? They clearly link directly to significant macroscopic health outcomes for the individuals, moving from the cellular level to the whole organism.
Precisely. This is where the profound implications become clear. These quantitative markers of genomic instability correlate directly and strongly with long-term health risks, particularly a significantly increased risk of various types of cancer decades down the line.
Decades later, wow.
Yes. It's a clear and powerful demonstration of how damage at the fundamental genetic level, the very blueprint of life, can translate into severe and life-altering disease. This isn't merely a correlation. It points to a direct causal pathway where the maintenance of genetic integrity is absolutely paramount for preventing serious pathologies and preserving health over a lifetime.
Now, here's where it gets truly fascinating.
Yeah.
It begins to connect directly to another core concept of aging, telomeres. How does an external insult like radiation exposure, which causes DNA damage, tie into the process of telomere erosion? Those protective caps on our chromosomes?
Well, the connection is profound, and it really illustrates the cascading long-term effects of genomic stress. Beyond just inflicting direct, immediate DNA damage, radiation exposure has been definitively linked to accelerated telomere erosion.
So the damage speeds up the shortening of the telomeres.
Exactly. Consider a powerful and sobering specific study that looked at the Chernobyl cleanup workers, often referred to as liquidators.
Right, I remember reading about them.
Even approximately 20 years after their initial acute exposure to high levels of radiation from the disaster, these workers had significantly and demonstrably shorter telomeres in their blood cells compared to carefully matched control groups who had not been exposed.
20 years later, that's incredible.
It is. This long-term shortening provides compelling evidence that the initial genomic insult set in motion processes of cellular aging that continued to manifest for decades.
That's a truly powerful long-term effect, indicating that the initial damage doesn't just resolve, but sets in motion insidious processes that continue to unfold for decades, impacting fundamental cellular structures. So what does this all mean for the bigger, overarching picture of aging? How do we integrate these insights into a coherent understanding of why we grow old?
This illustrates a critical and profound chain of events, what we can call the narrative arc of genomic instability and aging.
Okay.
Providing a comprehensive and interconnected framework for understanding this fundamental process. It begins with an environmental or endogenous insult, like ionizing radiation or even the simple byproducts of metabolism.
Things happening inside the cell, too.
Right, which leads to an increased DNA damage throughput, meaning damage is occurring at a much faster rate, or with significantly higher intensity and frequency than normal, healthy cellular processes can efficiently handle.
Overwhelming the system.
Precisely. This excess overwhelming damage then directly accelerates telomere attrition, which, as we'll delve into more deeply, is itself a key and well-established driver of cellular aging. The only thing that can effectively counteract this relentless risk, this continuous march of damage and degradation, is the presence of robust, highly efficient, and constantly vigilant DNA repair mechanisms within the cell.
The cell's maintenance crew.
Exactly. If these intricate repair systems are overwhelmed by the sheer volume of incoming damage, or if their inherent efficiency naturally declines with age, which it seems to do, the balance inexorably shifts towards genomic instability, leading directly to accelerated cellular and organismal aging. This paints a clear picture of the ongoing high-stakes battle within our cells to maintain genetic fidelity.
Okay, that first glimpse really sets the scene. We've seen how radiation causes direct DNA damage, leading to visible chromosome problems like aberrations and micronuclei.
And crucially, how that links to long-term health risks like cancer and even accelerates telomere shortening, connecting external damage to internal aging clocks. It really highlights the importance of DNA repair. Hashtag DNA damage modalities and repair pathways.
Okay, let's unpack this deeper, moving from the observable consequences of damage to the very types of damage our DNA experiences and how the cell attempts to fix them.
Right.
We've established that genomic instability happens when DNA damage simply outpaces the cell's capacity for repair. But DNA damage isn't just one monolithic threat, is it?
Not at all.
What are the different flavors or forms of damage we're talking about, and how does the cell's intricate machinery specifically address each one? It sounds like understanding the different ways a machine can break down.
That's a good analogy. You're absolutely right. DNA damage isn't a single, uniform entity. It comes in multiple distinct modalities or specific types. Each type possesses unique chemical characteristics, carries distinct consequences for the cell's function and viability, and critically is addressed by specific, highly evolved, and often remarkably sophisticated repair mechanisms meticulously designed to counteract that particular form of damage. The complexity is really quite astounding.
Let's start with the most common, the everyday wear and tear that our cells constantly experience.
Okay. The most frequent and pervasive type of DNA damage are single-strand breaks, SSBs.
Single strand, so just one side of the ladder.
Exactly. Imagine the DNA double helix as a twisted ladder. An SSB is precisely what it sounds like. A clean break in just one of those side rails, the sugar phosphate backbone.
How common are these?
Incredibly common. We estimate tens of thousands occur every single day within just one human cell.
Wow, that's a lot of patching needed.
It is. They are often caused by routine cellular metabolism, like the generation of reactive oxygen species, those free radicals.
Right, metabolic byproducts.
Yes. Or even ubiquitous low-level background radiation naturally present in our environment. Typically, SSBs are repaired with remarkably high accuracy and efficiency by highly specialized processes, such as base excision repair.
Base excision repair. Sounds specific.
It is. It's a highly efficient pathway specifically designed to correct damaged or modified individual bases and to relegate single-strand breaks.
So usually a quick fix, no big deal, almost like patching a small tear.
For the most part, yes, the cell is incredibly adept at handling these. The machinery is constantly patrolling and repairing. But here's the crucial catch.
There's always a catch.
Well, yes. If the repair of an SSB is delayed or becomes inefficient for any reason, maybe there's too much damage or the repair enzymes aren't working well.
Like with age.
Possibly. It can become problematic. For example, an unrepaired SSB can stall replication forks.
Stall the copying machine.
Exactly. Imagine the DNA replication machinery trying to move smoothly along the helix. If it hits this break, it grinds to a halt, halting DNA duplication for that segment.
That sounds bad.
It can be. Even worse, if left unrepaired, an SSB can spontaneously convert into a much more dangerous type of damage. A double-strand break.
Escalating the problem.
Profoundly. It moves from a minor repair job to a real cellular emergency.
Which brings us to the next flavor, double-strand breaks. DSBs. These truly sound like a far greater threat, like cutting the whole ladder in half.
They are indeed. DSBs are the most severe and perilous form of DNA damage, representing a complete severance of both strands of the DNA helix simultaneously.
Both strands gone.
Yes. This utterly disrupts the continuity of the genetic information. This is a profound threat to genome integrity because if not properly and precisely repaired, a DSB can lead to catastrophic consequences.
Such as
Large chromosome deletions, where a significant piece of the chromosome is permanently lost.
irreplaceable information.
Exactly. Or translocations, where a piece of one chromosome breaks off and inappropriately attaches itself to an entirely different chromosome. Think of it as chopping an instruction manual in half, or worse, mixing pages from different manuals.
Leading to chaos.
It can fundamentally alter gene expression, lead to severe cellular dysfunction, trigger cell death, or, perhaps most dangerously, contribute to cancerous transformation.
How do cells manage to tackle such an existential problem? They must have incredibly robust, almost emergency-level systems for this.
They do. Cells primarily rely on two distinct yet sophisticated main pathways to accurately fix DSBs, each with its own operational characteristics, advantages, and trade-offs.
Okay, what's the first one?
The first is non-homologous end, joining NHEJ. This pathway is remarkable for its readiness and speed. It operates throughout the entire cell cycle.
Always on call.
Pretty much. It acts like a rapid response team. Its mechanism is straightforward.
Right.
It works by directly ligating or rejoining the broken DNA ends, essentially gluing them back together.
Sounds incredibly fast, like a quick patch-up job.
It is indeed fast, which is its primary advantage, allowing swift containment of the damage. But its speed often comes at a cost. NHEJ is generally considered error-prone.
Error-prone. How so?
Because it doesn't use a template to guide the repair, it essentially brings the broken ends together and ligates them, even if they don't perfectly match up. This can lead to the introduction of small mutations, insertions, or deletions in DELs at the break site.
So it prioritizes speed over perfect accuracy.
Right. It prioritizes rejoining the backbone over absolute fidelity, which can lead to subtle genetic changes accumulating over time.
And what about the other pathway for DSBs? The high-fidelity option.
That would be homologous recombination, HR. This pathway is universally considered the high-fidelity repair pathway, much more accurate.
High fidelity, so it makes fewer mistakes.
Vastly fewer. However, this superior accuracy comes with a significant constraint. It's only active during specific phases of the cell cycle, specifically the S phase, when DNA is being replicated, and the G2 phase, after replication but before division. This is because HR crucially requires an intact sister chromatid as a perfect template.
So it leverages the cell's duplication process, using a perfect copy to precisely fix the broken one, like having a master blueprint right there.
Exactly, that's a perfect analogy. When a cell prepares to divide, it duplicates its DNA, creating two identical sister chromatids. HR brilliantly uses the intact sister DNA as a pristine template to accurately repair the break on the damaged chromatid.
Ensuring the original sequence is restored.
Precisely, without errors.
Yeah.
It's like having the architect's blueprint for flawless reconstruction. This makes it the preferred method when accuracy is paramount, like in germ cells.
Are there other types of DNA damage beyond just breaks? Things that might physically block the DNA's function?
Yes, absolutely. Another significant and particularly problematic modality is DNA crosslinks.
Crosslinks, like things getting stuck together.
Exactly. These are covalent links that form either directly between the two DNA strands themselves, physically preventing them from separating.
Which they need to do for replication and transcription.
Right. Or the crosslink can form between a DNA strand and an associated protein, essentially handcuffing the DNA.
Yeah.
These are often caused by environmental factors like intense UV light or certain chemicals, including some chemotherapy drugs.
And what's the impact of a crosslink? It sounds like a major roadblock.
You're exactly right. Crosslinks are problematic precisely because they physically prevent DNA strand separation, which is absolutely essential for replication and transcription. Without separation, these vital functions are blocked.
So the cell grinds to a halt.
It can lead to stalled processes, severe cellular distress, and ultimately, if unresolved, cell death. Specialized and highly complex repair pathways are needed to resolve them, involving intricate multi-step processes like nucleotide excision repair and sometimes complex forms of homologous recombination like the Fanconi anemia pathway.
So if these incredibly sophisticated mechanisms as entire repair crew fail or are overwhelmed, what's the ultimate consequence?
If DNA lesions escape repair altogether, or if they are misrepaired, especially by error-prone pathways like NHEJ, they can yield permanent mutations, lasting changes to the genetic code.
Or worse.
Or they can lead to larger-scale chromosomal instability, like those translocations we mentioned. These errors accumulate over time, and critically, over time, the relentless accumulation of such unrepaired or misrepaired damage is increasingly understood as a primary driver of aging.
The DNA damage theory of aging.
That's the central tenet. A lifetime of accumulating genomic imperfections contributes directly to the decline we associate with getting older.
So our cells possess this incredibly sophisticated, multi-layered repair network. But does this crew stay constant as we age? Does it get less efficient?
That's the critical question, and unfortunately, the research consistently indicates a trend. While cells have extensive repair networks, research suggests that with age, these systems, particularly the high-fidelity pathways like HR, may become notably less efficient, less active, or less accurately deployed.
So the balance shifts.
Exactly. This age-related decline tilts the balance. Instead of repair winning, damage accumulates more rapidly, pushing the cell further towards genomic instability and accelerating aging.
Okay, let's quickly recap the damage types and repair mechanisms. We have common single-strand breaks, usually fixed accurately by base excision repair.
But if delayed, they can stall replication or become double-strand breaks, which are much more serious.
Right. DSBs are severed chromosomes, fixed either quickly but imperfectly by NHEJ, or accurately but only sometimes by homologous recombination using a sister chromatid.
And then there are DNA crosslinks, physical blockages often caused by UV or chemicals needing complex repair pathways. Failure in any of these leads to mutations or instability.
And crucially, repair efficiency, especially for high-fidelity pathways, seems to decline with age, letting damage accumulate. That's a key part of the aging puzzle. Hashtag telomere biology and replicative senescence. Building on our understanding of DNA damage, let's now connect this to those fascinating protective caps on our chromosomes. Telomeres. What precisely are they, and how do they fit so intimately into the aging puzzle? They sound like the ultimate molecular clock.
That's an excellent way to put it. Telomeres are specialized, highly conserved, and profoundly important protective caps found at the very ends of our linear chromosomes.
Like the tips on shoelaces.
Exactly like the plastic or metal tips, the aglets. Their fundamental purpose is to prevent the main genetic material, the coding DNA, from unraveling, getting damaged, or fusing inappropriately with other chromosome ends.
Like it would be chaos.
Utter chaos. Without them, the ends would fray and stick together, causing catastrophic problems during cell division. In humans, these caps are made up of incredibly simple yet critical repetitive DNA sequences, specifically T-TAGI.
T-TAGI, repeated over and over.
Thousands of times, yes. Sometimes up to 15,000 base pairs at each chromosome end, forming a long, redundant stretch of protective DNA.
And they don't just sit there passively, right? There's a protein complex involved.
Correct. These repetitive DNA sequences aren't naked. They're intimately bound by a crucial 6-protein complex known as shelterin.
Shelterin, what does it do?
Its job is twofold and absolutely vital. First, it hides the chromosome ends. This is crucial because the cell's DNA damage sensors are constantly looking for break-in DNA ends.
Which telomeres could look like.
Exactly. Shelterin essentially tells the cell, these ends are normal, don't try to repair or fuse them. Secondly, shelterin physically prevents unwanted end-to-end chromosome fusions, which would be catastrophic.
So they protect the ends from being mistaken for damage. But you mentioned they shorten with cell division.
Right.
How does that happen? What's the mechanism behind this countdown?
This leads us directly to the end replication problem, a fundamental biochemical limitation inherent to DNA replication in linear chromosomes. Every time a cell divides, its DNA must be fully replicated. However, the enzyme responsible for copying DNA, DNA polymerase, simply cannot fully replicate the very 3R end of each linear chromosome.
Why not?
It's a fundamental biochemical constraint. It requires a short RNA primer to start synthesis, and when that primer is removed from the very end, there's no way to fill in the resulting gap on the lagging strand. This leaves a small, uncopied segment.
So with each and every cell division, we literally lose a small, consistent bit of that telomere length, like snipping off a tiny piece of the shoelace tip each time.
Precisely. It's a progressive, cumulative process in most somatic cells. After many rounds of division, maybe 40, 60 times for some human cells in culture, these telomeres erode to a critical, dangerously short length.
And then what?
At this crucial point, the shelterin complex can no longer effectively bind to and mask the chromosome terminus, exposing the unprotected DNA end.
So the cap is effectively gone. What does the cell perceive it as? Does it trigger the damage alarm?
Yes, absolutely. An uncapped, critically short telomere is immediately perceived by the cell as a double-strand break. DSB.
Even though it's a normal end?
Yes. The lack of shelterin causes the cell to misinterpret it as severe damage, triggering an urgent response.
This sounds like it triggers a major irrevocable alarm system in the cell, leading to a profound change in its fate.
It absolutely does, a profound and irreversible cellular response. In response to this perceived DSB at the telomere, the cell activates its sophisticated DNA damage response kinases.
Like ATM and ATR we mentioned?
Exactly, those master regulators. They, in turn, directly activate P53, the guardian of the genome.
Okay.
And P53 rapidly induces the expression of the cyclin-dependent kinase inhibitor P21.
And the ultimate outcome of this entire cascade, what's the final fate?
The profound and irreversible result is replicative senescence.
Senescence, meaning
a permanent cell cycle arrest. The cell stops dividing indefinitely, entering an irreversible growth arrest state. It's also famously known as the Hayflick limit, named after Dr. Leonard Hayflick, who first discovered human cells have a finite number of divisions.
this mechanism, this program stopping, sounds like it's there for a very good reason, a safety feature.
It is, unequivocally. Replicative senescence is widely considered a powerful tumor-suppressive failsafe.
How so?
By halting the division of old cells with short telomeres and potentially unstable genomes, it prevents them from proliferating uncontrollably, accumulating more mutations, and potentially turning cancerous. It's a critical barrier against tumorigenesis, especially early in life.
So it protects us from cancer.
Mm-hmm.
But then how does this very same protective feature paradoxically contribute to aging? It sounds like a biological trade-off.
It is a fascinating biological paradox, a classic example of antagonistic pleiotropy. While beneficial against cancer early on, telomere-driven senescence simultaneously contributes significantly to aging later by gradually exhausting the regenerative capacity of our tissues. If too many cells in a tissue like skin, blood cells, organ linings enter this permanent arrest,
The tissue can't renew itself.
exactly, that tissue loses its ability to repair, renew, or replace damaged cells. This progressive depletion leads to tissue and organ dysfunction, manifesting as the problems we associate with aging.
So if critical telomere shortening stops proliferation, does it also lead to wider genomic problems?
Yes, the consequences are multifaceted. Critical telomere shortening directly causes genomic instability. Those unprotected ends are vulnerable to breakage and fusion.
Right.
This resulting instability then triggers the response that halts proliferation, senescence, but can also trigger apoptotic death, programmed cell death, to eliminate severely damaged cells.
So either way, you lose functional cells.
All right, both outcomes reduce the pool of functional, dividing cells, diminishing the tissue's capacity for maintenance and repair.
And what about the enzyme that can extend telomeres, telomerase? Why isn't it active in most of our cells if it could prevent this?
Telomerase. The enzyme responsible for adding those TTG repeats back. It's normally silenced or expressed at extremely low levels in the vast majority of adult somatic cells, skin, liver, muscle cells, etc.
Why silenced?
This silencing is crucial. It ensures that progressive telomere attrition acts as that molecular clock, counting down cell divisions. It's part of that tumor suppressive mechanism, an intrinsic brake on uncontrolled proliferation.
So only a select few specialized cell types can maintain their telomere length.
Yes, precisely. Primarily stem cells responsible for tissue renewal and germ cells producing sperm and eggs express sufficient active levels of telomerase.
Why them specifically?
These cells need to divide extensively. Stem cells replenish tissues throughout life, and germ cells must transmit complete genetic information across generations without losing critical telomeric DNA. Telomerase itself is complex, with a protein part, TERT, and an RNA template, TERT.
Okay, so to recap telomere biology, they're protective chromosome caps made of T-TAGI repeats, shielded by the shelterin complex.
Right, but they shorten with each cell division due to the end replication problem. This acts as a molecular clock.
When they get critically short, they're seen as DNA damage, triggering a cascade involving P53 and P21, leading to replicative senescence, permanent cell cycle arrest.
This is a crucial anti-cancer mechanism, but paradoxically contributes to aging by depleting tissue regenerative capacity.
And most of our cells silence telomerase, the enzyme that extends telomeres, reinforcing this limit, while stem cells and germ cells keep it active for their specific functions. This sets up the whole dynamic of aging versus cancer risk. Hashtag genomic instability, senescence, and the SAP cascade. So a cell hits its Hayflick limit, goes senescent, stops dividing. But the consequences don't just stop there within that single cell, do they?
Not at all.
We're talking about a significant ripple effect, a way for these senescent cells to actively influence and even disturb their surroundings, creating a problematic environment.
Absolutely. When genomic instability or telomere dysfunction pushes a cell into senescence, the ramifications extend far beyond that single arrested cell. It impacts the whole tissue microenvironment, even systemic aging.
Oh, they don't just retire quietly.
Far from it. They actively adopt a dramatically altered gene expression profile known as the senescence-associated secretory phenotype, or SASP.
SAP, okay, what does that involve? What are these cells secreting?
The SASP involves the chronic, persistent secretion of a complex and potent cocktail of biologically active molecules.
Like what?
This includes various powerful inflammatory cytokines like IL-6 and IL-1 beta.
Pro-inflammatory signals.
Exactly. They also release specific chemokines which act like beacons to recruit immune cells, often worsening inflammation.
Okay.
Beyond that, they secrete growth factors that can alter neighboring cell behavior, sometimes promoting growth, sometimes suppressing it. And critically, they secrete proteases.
Enzymes that break things down.
Yes, enzymes that aggressively break down proteins, significantly remodeling and often degrading the extracellular matrix, the structural scaffold of tissues. It profoundly impacts tissue structure and function.
It sounds quite active and potentially disruptive, almost like a constant low-level irritant. But you mentioned SASP isn't always harmful.
That's a crucial nuance. SASP can indeed have beneficial roles, but typically in specific, tightly controlled short-term contexts.
Like wound healing.
Exactly. In acute wound healing, these secreted factors help coordinate tissue repair and remodeling, initiating a controlled inflammatory response to clear debris and help regeneration.
Makes sense.
It also helps recruit immune cells, like macrophages and NK cells, to eliminate precancerous senescent cells. So it acts like a surveillance system or cleanup crew. Short term, it can be beneficial.
But in aging, it sounds like it tips to the negative side, becomes chronic.
Precisely. This is where the balance shifts. In aging tissues, senescent cells accumulate persistently. They aren't cleared efficiently. So the chronic nature of SASP often does far more harm than good. This sustained secretion creates a pervasive, local, and often systemic environment of sterile inflammation without an active infection, driven by internal distress signals.
Right.
This also promotes extensive matrix remodeling, often leading to fibrosis or scarring. This persistent inflammatory state is strongly linked to various age-related pathologies.
Like heart disease, neurodegeneration.
Exactly. Atherosclerosis, Alzheimer's, type 2 diabetes, arthritis. It contributes to their progression. It transitions from a beneficial acute response to a chronically detrimental background state.
So what's the precise trigger for SASP? Does it stem directly from the genomic instability we started with?
Yes, that's the crucial connection. Genomic instability is unequivocally identified as a key upstream trigger. When DNA damage occurs, specifically persistent, irreparable double-strand breaks, or those uncapped critically short telomeres.
Things that cause senescence.
Right. It doesn't just halt division, it activates a sustained DNA damage response signaling cascade. This persistent signaling directly drives the production and secretion of SSP factors through several key pathways.
Which pathways?
Including the tumor suppressor P53, the pro-inflammatory transcription factor, NFB, and a complex called CGS-STING, which is an innate immune sensor for DNA in the cytoplasm. These pathways flip the switch for the SASP program.
Can you give a concrete example of how this plays out in experiments?
Certainly. In elegant studies using vascular endothelial cells, the cells lining our blood vessels.
Crucial for cardiovascular health.
Absolutely. Researchers simply disrupted the shelter-in proteins, effectively uncapping the telomeres. They found this single manipulation was sufficient to induce both senescence and a robust SSP-like phenotype.
Showing a direct causal link.
Exactly. Telomere dysfunction, a manifestation of genomic instability, directly triggers SSP.
And it sounds like this whole process can create a truly vicious cycle where the damage just keeps feeding itself.
It absolutely can, creating a detrimental, self-perpetuating feedback loop. We see a vicious cycle where oxidative stress and chronic inflammation fuel each other.
Yeah.
Oxidative damage can target telomeres or DNA, driving cells into senescence and triggering SSP. Then the CSP factors themselves, like IL-6 and IL-1, can cause further production of reactive oxygen species, ROS, and additional DNA damage in neighboring previously healthy cells.
A self-perpetuating cycle of damage and inflammation.
Exactly. A destructive cascade spreading through tissues.
So the accumulation of these senescent cells with their continuous SASP contributes to inflammaging. What does that mean for our tissues and overall aging?
Inflammaging refers to that chronic, low-grade, pervasive, sterile inflammation characterizing aging tissues, largely driven by senescent cells and their SSP.
And this contributes to disease.
Yes, this persistent inflammation is thought to be a significant contributor to overall tissue dysfunction and the development of numerous age-related chronic inflammatory conditions. We mentioned cardiovascular disease, neurodegeneration, metabolic disorders, musculoskeletal problems. It's a systemic background noise degrading tissue function.
So the crucial take-home point here is that genomic instability isn't just about cells dying or mutating directly. There's a broader, more insidious impact.
Exactly. The crucial message is that genomic instability doesn't just result in cell death or direct mutations. Even cells that survive but become senescent can propagate damaging signals far beyond themselves.
Through the SAP.
Yes, via the SAP, effectively poisoning or altering the microenvironment and spreading the detrimental impact to healthy neighbors. This contributes to and accelerates systemic aging processes. It's a broadcast of cellular distress affecting the whole system.
Right, so SAST is this cocktail of inflammatory molecules secreted by senescent cells, while it has short-term benefits like wound healing.
Its chronic presence in aging drives inflammaging, contributing to many age-related diseases.
And it's directly triggered by genomic instability, like irreparable DNA damage or short telomeres via pathways like P53, NF-kappa, and CGS sting.
Creating a vicious cycle where inflammation causes more damage, which causes more inflammation. It's not just about the senescent cell itself, but its damaging effect on the whole tissue environment.
This idea of SATP spreading damage is fascinating. And now we're diving into an even more intriguing recent discovery about how genomic instability directly feeds into ATP through something unexpected: jumping genes.
Yes, this is a truly groundbreaking area. An unexpected mechanism linking genomic instability to the SAP cascade was uncovered by recent studies focusing on retrotransposons, often called jumping genes.
Jumping genes, what are those? They sound like something ancient in our DNA.
They are. Retrotransposable elements, like the well-known line 1, L1 elements, make up a surprisingly large portion of our DNA, maybe up to 17% of the human genome.
Wow, that much.
Yes. In healthy young cells, these elements are typically kept silenced, locked down, by intricate epigenetic mechanisms. They're usually dormant.
Epigenetic silencing keeps them quiet. But something changes in senescent cells.
Indeed. In senescent cells, these epigenetic controls often fail. This process is called de-repression. This means these L1 elements become transcriptionally active, they get read and copied into RNA, they wake up.
And then they can literally copy and paste themselves around the genome.
Precisely. These L1 sequences transcribe into RNA, then use a special enzyme they encode, a reverse transcriptase, to convert that RNA back into a DNA copy called cDNA.
RNA back to DNA.
Yes. This L1 cDNA can then integrate itself elsewhere in the genome, literally jumping to a new location. This reintegration itself can be mutagenic, disrupting genes or creating new breaks, adding to instability.
So these newly copied L1 cDNAs are floating around, maybe even outside the nucleus. How does that trigger inflammation? It's our own DNA, but it's out of place.
This is the key insight. The buildup of this cytoplasmic L1 cDNA, L1 DNA copy is accumulating in the cell's cytoplasm outside the nucleus, is sensed by the cell as foreign or dangerous DNA.
Like a virus.
Exactly. It's as if the cell detects a viral infection, even though the material is endogenous. This triggers a robust, innate antiviral inflammatory reaction, specifically activating a type I interferon, IFNI, response, often via that CGS sting pathway we mentioned earlier.
An innate antiviral response, even though it's our own DNA. A profound viral mimicry.
Yes, precisely. And this activated IFNI response, in turn, significantly reinforces and amplifies the SSP, directly contributing to the chronic inflammation in aged tissues. This interferon-driven SSP seems to be a hallmark of late-stage senescence, a powerful amplification loop.
This sounds like a prime target for intervention. If you stop this jumping, this retrotransposition, can you dampen the inflammation?
Remarkably, recent landmark studies showed exactly this. If you block the L1 reverse transcriptase activity, you can significantly dampen this whole inflammatory process.
How did they do that?
Researchers achieved this by treating old mice with a well-known drug called lamivudine.
Lamivudine. Isn't that an HIV drug? Incredible repurposing.
It is indeed. Lamivudine is a nucleoside reverse transcriptase inhibitor, RTI, widely used for HIV and hepatitis B. It blocks the viral reverse transcriptase. By repurposing it, researchers found it profoundly blunted the IFN-I activation and strikingly reduced age-related inflammation across multiple tissues in old mice.
So shutting down retrotransposon activity pharmacologically actually alleviated inflammaging. This is huge.
It was indeed a groundbreaking clue. It strongly suggested that retrotransposition-induced inflammation is a significant driver of aging and, crucially, represents a novel and potent therapeutic target.
Was the effect visible?
Yes, the visual evidence is quite striking. If you look at fluorescent microscopy images of fat tissue, a young mouse shows minimal inflammatory cells. An old, untreated mouse exhibits numerous infiltrating inflammatory cells, glowing pink or magenta. But after just a short course of lamivudine, those old mice show markedly fewer inflammatory cells. A clear visual demonstration.
Beyond just reducing inflammation, what were the broader functional benefits in these mice? Did their health improve?
Yes, these findings, while still early stage, truly open the door to repurposing RTIs for age-associated disorders. Lamivudine in aged mice not only dialed down inflammatory cytokines, but also showed tangible functional benefits.
Like what?
For example, 6 months of treatment in 20-month-old mice, quite old, led to measurable improvements like reduced muscle and fat loss, sarcopenia, and significantly less tissue fibrosis or hardening.
And a crucial point. Did lamivudine reverse senescence or lengthen telomeres, or was it purely downstream?
No, and this is key. Lamivudine explicitly did not reverse senescence or lengthen telomeres. What it did was simply quiet one major downstream consequence, the IFN-SASP feedback loop. It neutralized one of senescence's most damaging effects.
So even if the initial damage can't be prevented or reversed by this drug, mitigating the inflammatory echo can still yield benefits. Very clever.
Exactly. It suggests that even if primary DNA damage or telomere erosion can't be fully reversed, mitigating the powerful echo, the inflammatory SP might still yield significant health benefits. It opens up a different, perhaps more accessible therapeutic strategy. Manage the fallout.
Are there any human trials yet for this intriguing approach?
There are very early translational efforts underway. A small Phase 2A open-label pilot trial involved 12 patients with mild cognitive impairment, a risk state for Alzheimer's.
Okay.
In this trial, 6 months of lamivudine treatment was found to be safe and was associated with reduced inflammation and improvement in 2 specific biomarkers of neurodegeneration.
That sounds promising, particularly for neurodegeneration. But I imagine there are significant caveats.
Yes, absolutely. These human data are preliminary and must be interpreted with extreme caution. It was open-label, lacked a placebo control, and the sample size was tiny, so many limitations.
Right.
However, despite these limitations, they do provide a tantalizing hint that taming retrotransposon activity could represent a novel therapeutic angle in age-related diseases, especially those with neuroinflammation. Larger, more robust trials are being planned.
And just to reiterate, this approach isn't fixing the initial problem, right? It's intervening later.
No, it's vital to reemphasize that. This targets a consequence of genomic instability, the viral mimicry and resulting inflammation, not the DNA damage itself or telomere shortening. So it would likely be complementary to other interventions aimed at the root causes, part of a multi-pronged strategy.
This section on retrotransposons was fascinating. So these dormant jumping genes, like line 1 elements, get reactivated in senescent cells due to failing epigenetic controls.
Right. They copy themselves via RNA and reverse transcription, creating DNA copies, cDNA, that accumulate in the cytoplasm.
Which the cell detects as foreign or viral, triggering a potent type I interferon response, a kind of viral mimicry.
Exactly. And this interferon response strongly amplifies the SSP, driving chronic inflammation or inflammaging from within.
And incredibly, repurposing the HIV drug lamivudine, an RTI, blocks this process in mice, reducing inflammation and improving health markers without reversing senescence itself.
Yes, targeting a major downstream consequence. Early human data is cautious, but hints at potential, needing much more research. It's a really novel angle on tackling age-related inflammation.
So given everything we've discussed about telomere attrition being a fundamental driver of aging, the most intuitive solution might seem to be just activate telomerase, right?
Yeah, it seems obvious.
Make telomeres longer. But this is precisely where we encounter a major biological yin-yang, a powerful and potentially dangerous duality.
It's indeed a very tempting strategy. If shortening telomeres cause aging, why not just lengthen them? Activating telomerase is undeniably a powerful tool. In cell culture, simply introducing the telomerase enzyme can immortalize cells.
Make them divide forever.
Essentially, yes, allowing them to divide far beyond the normal Hayflick limit.
And in living organisms, has this shown promise? Has anyone successfully extended lifespan in animals by activating telomerase?
Yes, telomerase interventions have shown truly remarkable promise in extending healthy lifespan in some organisms, with very important caveats. A landmark study in 2012, led by Maria Blasco, delivered the telomerase gene, TERT, into adult and aged mice using an AAV gene therapy vector.
What were the results in these mice? Did their health improve?
The treated mice showed truly remarkable and widespread health improvements. Better glucose tolerance?
Metabolic health?
Yes. Less osteoporosis, indicating stronger bones, improved neuromuscular coordination, and numerous other signs of rejuvenation across various organ systems.
And crucially, how did it affect their actual lifespan? Did they live longer?
Not only did their health span improve dramatically, but their median lifespan was extended significantly. Mice treated at 1 year of age saw about a 24% longer lifespan. Even those treated at 2 years quite old for a mouse showed around a 13% increase.
Those are substantial extensions. Now, here's the big question, the elephant in the room.
Yeah.
Cancer. Did these mice get more cancer?
This is the crucial finding from that study, the one that really shifted the paradigm. These telomerase-treated mice did not suffer an increase in cancer incidence compared to controls.
No increase. That's huge.
It was hugely significant, a proof of principle. It suggested that short-term controlled telomerase therapy in normal adult mice could delay aging without immediately unleashing rampant cancer. It boosted the idea that enhancing telomere maintenance could genuinely enhance longevity safely, at least in this model.
So if it didn't increase cancer in these mice, why is telomerase activation still called a double-edged sword? Why the persistent caution?
The reason is the deep, extensive understanding of telomerase's critical role in human cancer progression. Uncontrolled, sustained telomerase activity could enable incipient cancer cells already on the path to cancer, to bypass the mortality normally imposed by telomere shortening.
It removes a natural break?
Exactly, a critical safeguard.
What does that mean specifically in human cancers?
Well, about 85–90% of all human cancers reactivate telomerase. It's a critical step for them to achieve immortality, that uncontrolled, indefinite proliferation.
So it's essential for most cancers?
Yes. The natural telomere shortening in our normal cells is a powerful anti-cancer mechanism. Would-be tumor cells that proliferate excessively hit that telomere barrier or a crisis checkpoint and should stop or die. If telomerase removes that barrier, they can keep dividing indefinitely, potentially forming tumors if other mutations are present. Telomerase becomes an on switch for pathological growth.
So any therapy boosting telomerase carries a theoretical but significant risk of promoting cancer.
Precisely. Any telomerase-enhancing therapy inherently carries a theoretical oncogenesis risk. It's a classic biological trade-off. More regeneration versus higher cancer risk. A very high-stakes balance.
Given this link to human cancer, why do you think that mouse study didn't show increased cancer? Any specific reasons?
Several factors likely contributed. First, the therapy started in adult or old mice, which typically have fewer actively dividing stem cells prone to transformation compared to younger animals. Older organisms also generally have more robust tumor surveillance. Second, and critically, the telomerase expression via AAV wasn't indefinite, it tapered off. This transient activation might have been enough for benefits without pushing cells into sustained, uncontrolled proliferation needed for cancer.
What does this mean for potential human application? How do we balance this?
In humans, the risk envelope might be much narrower. Even a small theoretical chance of accelerating a hidden precancerous lesion could be unacceptable for a general anti-aging therapy. Nonetheless, researchers like Maria Blasco are actively investigating ways to transiently or tissue-specifically activate telomerase. The goal is to get the benefits without the tumorigenic risk.
How could that work?
Future therapies might involve tightly regulated boosts, maybe an inducible gene therapy you can turn on and off, or intermittent drugs combined with rigorous monitoring.
So it really is the ultimate geroscience yin-yang. Immense promise, significant peril.
Yes, telomerase activation perfectly exemplifies this. It holds immense promise for reversing cellular aging and significant peril for promoting cancer. Any translation into humans would demand non-negotiable oncology surveillance.
Meaning what, exactly?
Frequent comprehensive screenings, periodic whole-body imaging, highly sensitive blood tests for circulating tumor DNA, ctDNA, to detect cancer very early. Vigilance is key because dormant precancerous cells might exploit telomere elongation.
Yeah, end of science peer.
Immediate intervention. Stop the therapy, maybe use senolytics or cytotoxic drugs to eliminate emerging problems.
And who would even be eligible?
Eligibility would likely be very tightly controlled initially, excluding patients with prior cancer or premalignant conditions, maybe combining telomerase therapy with anti-cancer safeguards.
They're not a general anti-aging pill.
Unlikely soon. As it stands, telomerase might best be harnessed for clear, severe medical needs. Rare premature aging syndromes like dyskeratosis congenita or idiopathic pulmonary fibrosis diseases linked to short telomeres. Or maybe localized tissue engineering.
Could we make it more targeted? Only activate it where needed.
That's an active and critical area of research. Could we target it to specific cells, like hematopoietic stem cells, to rejuvenate the immune system, while minimizing exposure in epithelial cells prone to cancer?
Like gut lining or skin.
Exactly. These are critical questions that must be resolved before telomerase rejuvenation can be safely attempted in humans. Precise control is paramount.
Okay, this look at telomerase really highlights the complexity. Activating it showed amazing promise in mice for health span and lifespan without increasing cancer in that specific study.
But its undeniable role in enabling most human cancers makes it a true double-edged sword. Any human use carries a profound oncogenesis risk.
Meaning strict, non-negotiable cancer surveillance would be essential, along with highly controlled, likely targeted or transient delivery, probably restricted to severe medical needs initially, not general aging. It's a potent but dangerous tool. We've discussed how genomic instability triggers chronic inflammation via SSP and how telomere erosion impacts cell division. But genomic damage isn't isolated, is it? It also seems to profoundly affect our cells' fundamental energy systems.
You've hit on a crucial point. Genomic instability isn't isolated. It deeply perturbs and intricately interconnects with cellular metabolism. A key player in this critical metabolic crosstalk is an enzyme called PRP1.
Poly-ADP ribose polymerase 1, what does it do?
PRP1 is essentially a highly active DNA damage sensor and a crucial repair enzyme. It specifically detects single-strand breaks and various other DNA lesions.
Like a cellular patrol finding damage.
Exactly. When it finds damage, PRP1 responds by rapidly adding long chains of ADP ribose onto itself and other nearby proteins involved in repair. This process, called pyrrolation, acts as a critical molecular signal.
A flare signal.
Kind of. It helps recruit the necessary DNA repair, machinery ligases, polymerases, chromatin remodelers to the exact site of damage, guides the repair team.
So it's definitely a good guy, essential for repair. But you mentioned a significant metabolic cost. A downside.
This is precisely where the metabolic crosstalk comes in, exposing a fundamental trade-off. PAP1's enzymatic activity, adding those ADP ribose chains, consumes a vital molecule called NAD plus SET.
NAD plus SWAG, nicotinamide adenine dinucleotide. That's crucial for energy production, right?
Absolutely crucial. NAD plus acts as a coenzyme in hundreds of metabolic reactions, especially in mitochondria for energy production. It's also vital for other important enzymes, particularly sirtuins.
So if there's a lot of DNA damage, PRP1 gets really busy. What happens to NAD plus LEG?
Exactly. When DNA damage is abundant and persistent, PAP1 can become chronically hyperactivated. This excessive activity leads to a rapid and significant depletion of the cell's critical NAD plus reserves.
Like draining the fuel tank for emergency repairs.
Precisely. It's like a vital resource is being siphoned off, leaving less available for other essential cellular functions. The cell's energy budget gets strained.
And what are the downstream effects of low NAD plus taro if this crucial molecule is exhausted?
This NAD plus depletion can trigger a cascade of detrimental effects. Low NAD plus means sirtuin deacetylase enzymes like SIRT1 become less active because they need NAD plus to work.
And SIRT1 is important for?
SIRT1 is crucial for many aspects of health and longevity, metabolic regulation, DNA repair itself, inflammation control. Its inactivity, linked to this NAD plus drain, is strongly correlated with severe mitochondrial dysfunction.
Powerhouse is not working well.
Right, and a general metabolic decline, which are hallmarks of aging.
Is there concrete scientific evidence of this chain? DNA damage, TRTRP plus depletion, sirtuin issues aging phenotypes.
Yes, very clear evidence. Research in progeroid models, those animals with premature aging, showed that cells deficient in DNA repair had chronically overactive PRP1. This led directly to NAD+ depletion, which disabled SART1 activity and severely disrupted mitochondrial function. It was a clear demonstration of this destructive feedback loop, mimicking and accelerating aspects of normal aging.
So this implies a potential therapy. Can you restore NAD+ or inhibit PRP1 to break this cycle?
Remarkably, yes. Treating these progeroid models with NAD+ boosters, like NMN or NR.
Nicotinamide mononucleotide or riboside?
Yes, precursors that replenish cellular NAD+ FINR, or by using PRP inhibitors, drugs that block PRP1 activity. Both approaches were able to restore SIRT1 activity and significantly improve mitochondrial health.
Suggesting intervening here can counteract age-related dysfunction.
Exactly. It provides compelling evidence.
This strongly implies that some aspects of aging, like fatigue and metabolic slowdown, might stem from this constant NAD drain caused by accumulated DNA damage.
It strongly suggests precisely that. A significant part of the aging phenotype might indeed stem from this NAD drain caused by the cumulative burden of DNA damage.
Because in normal aging, NAD+ declines and DNA damage increases.
Correct. We observe both. The logic is that chronic, low-level DNA damage continuously siphons NAD+ away for PRP1 repair at the expense of other vital functions like metabolic regulation via sirtuins.
So the connection is reciprocal. DNA damage affects NAD+ board, and NAD+ levels affect repair. A circular problem.
Exactly. This DNA damage NAD+ crosstalk is critically important and bidirectional. Robust DNA repair needs plenty of NAD+ bearer, but too much damage can bankrupt the NAD+ budget, impairing other essential functions. It's a vicious cycle.
This sounds like an incredibly promising therapeutic avenue, targeting a core metabolic vulnerability.
It does have significant therapeutic implications, hence the intense research. Approaches like NAD+ supplementation or maybe mild PRP inhibition might ameliorate some aspects of aging by breaking this cycle.
But there are cautions.
Yes, caution is warranted. While NAD+ boosters show positive effects in aged animal models, indiscriminately inhibiting PRP1 could paradoxically reduce overall DNA repair capacity since PRP1 initiates repair.
Which could be harmful long-term, allowing mutations.
Potentially, yes.
So what's the ideal strategic approach here? A delicate balance.
The ideal strategy might not be to suppress PRP1 outright, but rather to ensure cellular NAD+ levels are maintained optimally. Perhaps through precursors like NR or NMN or lifestyle means supporting NAD+ synthesis.
Golby.
To provide sufficient NAD+ so PRP1 can work effectively for repair without depleting reserves, and so sirtuins can function optimally for metabolic regulation and other processes. This interplay between genomic instability and energetics shows how deeply interconnected aging hallmarks are. Genome damage reverberates into metabolic decline, which can feed back into more damage. A complex web.
Okay, a quick recap on the NAD+ connection. PRP1, a key DNA repair enzyme for single-strand breaks, uses NAD+ as fuel.
Right. When DNA damage is high, PRP1 becomes hyperactive and depletes cellular NAD+ white.
This NAD+ depletion then cripples other NAD-dependent enzymes like AKRT1, leading to mitochondrial dysfunction and metabolic decline, core features of aging.
And it's a two-way street, as low NAD+ can also impair efficient repair. Boosting NAD+ or carefully modulating PARP shows promise in breaking the cycle in models, but needs careful handling. It really links our genetic integrity directly to our energy metabolism. #tag practical measures to reduce genomic instability.
We've gone incredibly deep into the molecular mechanisms and potential future therapies, but for the person listening, the natural question is any of this inevitable? Are there practical steps we can take right now to actively reduce genomic instability?
That's an excellent and highly practical question, moving from theory to action. And the answer is a resounding yes.
Good.
Not all genomic instability is inevitable. There are concrete, actionable steps, often termed clinical hygiene or preventative measures, that individuals and healthcare systems can implement to minimize avoidable DNA damage throughout life.
Proactively slowing things down.
Exactly, thereby slowing the pace of biological aging and extending health span.
What's a big one that comes to mind immediately? Something maybe related to skin?
One of the most major, widely applicable recommendations is UV avoidance and meticulous protection. Ultraviolet radiation from sunlight is incredibly potent and pervasive.
Everywhere.
Yes, a major source of DNA damage, particularly in skin cells. UV light causes specific lesions like cyclobutane pyrimidine dimers, CPDs.
Like those abnormal bonds.
If these aren't repaired, they can lead directly to mutations. That's why skin cancers are so common. And beyond cancer, UV exposure directly accelerates skin aging, not just wrinkles, but telomere shortening and genomic instability in skin cells.
So practicing sun-safe behavior isn't just cosmetic, it's fundamental DNA protection.
Absolutely not just cosmetic. Practicing sun-safe behavior using broad-spectrum sunscreen, wearing protective clothing, avoiding intense midday sun directly reduces cumulative UV damage. It affects fundamental genomic integrity, a direct, everyday intervention.
What about infectious agents? Can some common infections contribute?
Another critical measure is vaccination against oncogenic viruses. Certain viruses drive genomic instability and cancer. A prime example is human papillomavirus, HPV.
HPV and cervical cancer.
Right. Chronic HPV infection leads to DNA damage, telomere dysfunction, and chromosomal instability because HPV oncoproteins disable the cell's natural repair checkpoints and tumor suppressors like P53.
And other viruses.
Similarly, vaccination against hepatitis B virus, HBV, is crucial for preventing liver damage and cancer. HBV causes liver cancer through chronic inflammation and oxidative DNA damage.
So vaccines protect our genome too?
Effectively, yes. By vaccinating against HPV and HBV, we prevent these specific infectious sources of genomic stress, heading off downstream cancers and related damage. A highly effective preventative strategy.
What about general lifestyle habits? Diet, substances?
Maintaining healthy lifestyle habits pays significant dividends. For example, rigorously avoiding tobacco smoke is paramount.
Full of mutagens.
Exactly. It inflicts profound damage on lung cells and other tissues. Moderating alcohol intake is also important. Heavy drinking can lead to acetaldehyde-induced DNA crosslinks, a nasty form of damage.
And diet.
While complex, eating antioxidant-rich foods like colorful fruits and vegetables may help counteract some oxidative DNA damage by neutralizing reactive oxygen species.
On the medical side, how do clinicians minimize damage during procedures?
In medical settings, clinicians meticulously adhere to the Alivare principle as low as reasonably achievable for imaging involving radiation, like CT scans or X-rays.
Use the lowest dose possible.
Right. Unnecessary scans are avoided. Necessary ones use the lowest dose for clear image. Similarly, in radiation therapy for cancer, techniques are constantly refined to minimize exposure to healthy tissue. These practices aim to minimize iatrogenic DNA damage caused by medical intervention.
Recognizing every bit adds up.
Exactly. Each bit of excess damage can cumulatively contribute to aging or secondary cancer risk.
And something often overlooked but key, chronic inflammation.
Absolutely. Diligent infection control and inflammation management is vital. Chronic inflammatory states, whether from untreated infections like gum disease or chronic conditions like arthritis,
Bathe tissues and damaging molecules.
Yes, reactive oxygen species, ROS, and pro-inflammatory cytokines. These directly inflict DNA damage and erode telomeres. So, promptly treating chronic infections and effectively managing inflammatory conditions with meds or lifestyle changes can significantly preserve genomic integrity.
So it sounds like while we're waiting for high-tech interventions, there's a lot we can do with old-fashioned prevention.
That's precisely the summary and the empowering message. While high-tech interventions are exciting, much can be achieved through fundamental old-fashioned prevention and health management. The goal is to avoid or mitigate known DNA insults to slow the ticking rate of damage accumulation, thereby extending health span. It's about proactive biological stewardship.
Right, so practical steps are definitely possible. Top of the list, UV protection, sunscreen, clothing, avoiding midday sun.
Vaccination against oncogenic viruses like HPV and HBV is also crucial.
And lifestyle. Definitely no tobacco, moderate alcohol, and likely benefits from antioxidant-rich foods.
Clinically, minimizing radiation exposure via the Alivare principle and actively managing chronic inflammation and infections are key strategies, too. It really emphasizes prevention as a powerful tool for genomic health. Hashtag biomarkers of DNA damage and telomere attrition.
This entire field relies heavily on precise measurement. How exactly do scientists and clinicians quantify genomic instability and telomere attrition? What are the specific validated biomarkers they use?
That's critical. The ability to accurately measure is essential for understanding, tracking, and knowing if interventions work. A variety of robust, sensitive, and well-validated biomarkers are routinely used.
Let's start with a direct marker of those dangerous double-strand breaks. How do they count them?
Okay, first we have H2AX foci. When a DNA double-strand break, DSB, occurs, a specific histone protein, H2X, near the break gets phosphorylated, chemically modified.
To become H2AX.
Exactly. These marks can be visualized under a fluorescence microscope as distinct glowing spots foci within the nucleus using specific antibodies.
So you can count the spots.
Yes, they serve as an incredibly sensitive and quantitative indicator of DSBs. Researchers count these foci as a direct readout of DNA damage. Elevated OH2AX levels are consistently seen in aging cells.
Indicating more damage.
Right. And even one persistent focus can be enough to trigger senescence. In occupational studies, radiation workers show significantly higher foci counts, reflecting ongoing damage.
And you mentioned micronuclei earlier. How are those used as biomarkers?
Micronuclei are another classic cytogenetic marker. As we described, they're tiny, separate nuclei formed from chromosome fragments or whole chromosomes lost during cell division.
Errors in mitosis.
Yes. Their presence, especially in peripheral blood lymphocytes, is a strong indicator of genomic instability. Higher counts are consistently associated with radiation exposure, about 1.4-fold higher in workers and with aging generally.
And they have other effects too.
Yes, their presence can stimulate that CGA sting innate immune signaling, treating the mislocalized DNA as a danger signal, potentially contributing to SSP.
How about telomere length itself? That seems like a direct measure of the molecular clock.
Indeed. Telomere length and fragility are crucial biomarkers. Telomere length can be measured using various sophisticated techniques.
Like qPCR.
Yes, qPCR gives a relative length. FlowFush allows measurement on individual chromosomes. And the gold standard Southern blot TRF analysis gives a very accurate average length.
Is the average length the whole story?
Not always. Researchers often look at the distribution of lengths. Aging and stress typically shift the distribution towards shorter telomeres and increase heterogeneity, a wider range of length, indicating instability.
So telomere attrition, the shortening process, is a direct biomarker of cellular aging.
Yes, telomere attrition is considered a key biomarker. Leukocyte telomere length tends to progressively shorten with age and has been weakly linked to age-related disease risk, though individual variation is large.
And radiation affects it.
Yes, studies show an inverse correlation with lifetime radiation dose, more exposure, shorter telomeres. Also, telomere fragility, detected as breakage in certain assays, reflects replication stress at the ends. Measuring length distributions before and after therapy is crucial for evaluating interventions.
What about broader chromosomal changes, large-scale rearrangements?
Then there are chromosomal aberrations. Classical karyotyping staining and viewing chromosomes, or newer techniques like spectral karyotyping using colored dyes.
Right, making swaps obvious.
And whole genome sequencing can reveal large-scale aberrations, translocations, deletions, aneuploidy, abnormal chromosome number. These accumulate with age.
Like in bone marrow.
Yes, older individuals often show higher rates of cells with translocations or trisomies there. Clonal chromosomal abnormalities in blood, known as clonal hematopoiesis, increase significantly with age and raise blood cancer risk. Radiation workers definitely show more breaks and translocations.
Is there a simpler measure?
A related surrogate is sister chromatid exchange, SCE frequency, measured by differential staining. More SCEs can indicate instability or past genotoxic exposures.
And finally, beyond measuring damage, can we assess how well the cell can repair itself, its capacity?
Indeed. While not a direct marker of existing damage, assays measuring DNA repair capacity markers provide vital functional insights.
Like the COMET assay.
Yes, the COMET assay damaged DNA migrates out forming a tail. Longer tail means more damage. Or host cell reactivation assays measure how well cells repair a specific lesion in a reporter gene. These serve as excellent functional biomarkers of the cell's ability to handle damage, and these capacities often decline with age.
So, are these sophisticated biomarkers routinely used in clinics?
Currently, mostly in research or specialized testing, not routine checkups. However, they are invaluable in gerontology studies and essential for evaluating new interventions.
To see if a treatment actually works at the molecular level.
Exactly. Does it reduce SOK H2X foci? Slow telomere shortening. Improve repair capacity. In the future, one could envision a comprehensive panel becoming part of an aging clock to assess biological age and track outcomes.
Any progress towards clinical use?
Well, a 2021 meta-analysis highlighted chromosome aberration and micronucleus assays as consistently elevated in radiation workers, recommending them as standard biomarkers for monitoring such high-risk groups. Large studies are also tracking these markers over time to see how well they predict health span.
Okay, so measuring genomic health is key. We've got markers like H2AX foci for double-strand breaks and micronuclei for mitotic errors.
Precise measurements of telomere length distribution and fragility track the molecular clock.
Larger scale issues are seen with chromosomal aberration analysis, like karyotyping or sequencing.
And importantly, functional assays like the COMET assay measure the cell's actual DNA repair capacity, which often declines with age. While mostly research tools now, they're vital for understanding aging and testing interventions.
We've explored some incredibly promising potential interventions, from repurposing drugs to cutting-edge gene therapies. But with such powerful tools, there must be profound considerations around safety, right?
Absolutely critical.
We can't just unleash these therapies without incredibly careful thought and testing.
You've hit the most critical point for any translational science, especially with something as fundamental as aging. Safety monitoring and meticulous risk management are paramount. As we explore interventions targeting genomic instability in telomeres, it's clear a meticulous approach is required.
So scientists are developing ways to weigh the pros and cons.
Yes, scientists, clinicians, and regulators are actively developing and applying risk matrices and frameworks to weigh the potential benefits against the inherent risks. It's essential for responsible progress.
What are the main benefits and risks we're balancing?
On the benefit side, potentially extended health span, preventing age-related diseases, maybe even genuine rejuvenation, transformative potential.
And the risks?
The major concerns include promoting cancer and unforeseen long-term side effects. This applies especially to strategies like telomerase activation, but even long-term use of RTIs like lamivudine needs careful consideration.
Let's start with telomerase therapies, the double-edged sword. What does non-negotiable oncology surveillance mean in practice?
For telomerase-based therapies, the consensus is rigorous oncology surveillance would be an absolute prerequisite, non-negotiable.
Meaning?
Patients would need frequent, comprehensive screenings for early signs of cancer, periodic whole-body imaging, maybe PT-CT or MRI, highly sensitive blood tests for circulating tumor DNA, ctDNA.
Detecting cancer DNA shed into the blood.
Exactly, long before symptoms appear. This vigilance is crucial because dormant precancerous cells, which many people harbor, might seize the opportunity of telomere elongation to progress rapidly.
And if signs of abnormal growth appear?
Immediate, decisive intervention. Maybe stopping the therapy, perhaps using senolytics to kill senescent cells, or cytotoxic drugs to cull emerging cancerous clones before they become established tumors.
What about controlling who gets telomerase therapy and how it's delivered?
The who and how must be meticulously controlled. Maybe limiting it to older patients where regenerative needs are greater and cancer surveillance is routine, or only for specific diseases where benefits clearly outweigh risks.
Like those premature aging syndromes.
Yes. Furthermore, telomerase might be delivered temporarily or targeted. Gene therapy with a tissue-specific promoter, so only certain organs get the boost, or a drug activating it transiently in specific stem cells. These layers of control are critical for the risk matrix.
I also recall the concept of a suicide gene, an extra safety layer.
Yes, a very advanced safeguard. Integrated into the gene therapy vector. It allows the therapy to be switched off or the cells expressing it killed if something goes wrong or a cancer signal appears. An internal emergency brake.
Now shifting to the reverse transcriptase inhibitor strategies like lamivudine for L1 suppression, their risk profile seems more favorable.
Yes, for RTI strategies targeting L1, the risk profile appears significantly more favorable. Lamivudine is FDA approved with decades of safety data from HIV patients.
Right.
It doesn't permanently alter cells, and known side effects are relatively manageable.
But there are still unknowns with long-term use, specifically in aging populations.
Absolutely. Even here, caution is advised. The risk matrix must account for unknowns. Could long-term RTI use have off-target effects on other cellular polymerases, like mitochondrial ones?
Some older RTIs did cause mitochondrial toxicity.
Exactly. Could suppressing retrotransposons have unforeseen negative consequences, affecting beneficial tissue repair or even neuronal plasticity involved in learning?
Good point.
And if used broadly in healthy aging people, might it foster drug-resistant HIV in those with undiagnosed infections? Speculative concerns, but crucial points for monitoring in trials.
So close monitoring will be key for these trials too, even if the risk seems lower.
Indeed. While lamivudine progresses into trials for age-related conditions, participants will be closely monitored for adverse effects, immune changes, unexpected pathologies. The pilot study showed good safety over 6 months, but larger, longer trials are essential for long-term risk assessment.
In essence, scientists and regulators are meticulously building a comprehensive framework for these new therapies.
Exactly. In building a robust risk matrix, they enumerate all such considerations to decide on safeguards. For telomerase gene therapy, maybe lifelong annual oncology screening, integrating a suicide gene.
And for RTIs,
Maybe periodic blood tests for inflammation markers to ensure desired effects persist, and crucially, that immune surveillance of real pathogens isn't impaired over time.
and the risk level changes depending on the patient's health, right? Healthy aging versus severe disease.
Yes, a fundamental consideration. The bar for safety is significantly higher if treating healthy aging versus a life-threatening condition.
So initial use will likely be targeted.
Highly probable. Telomerase or RTI interventions will likely find initial use in specific diseases, idiopathic pulmonary fibrosis, certain neurodegenerative diseases where the mechanism is relevant. This approach justifies the risk by the disease context and, crucially, gathers invaluable safety and efficacy data to inform safer, broader applications later if the evidence supports it. #outro.
What an incredible, intricate, and deeply insightful journey we've taken into the core mechanisms of aging.
It really has been.
We've seen how genomic instability and telomere attrition aren't just abstract concepts, but powerful forces shaping our health, influencing everything from cancer risk and inflammation to metabolic well-being.
Indeed. Our rapidly growing understanding of this complex web has truly opened transformative doors to exciting potential interventions, from repurposing existing drugs to quiet genomic noise, like lamivudine suppressing line 1 elements.
Yeah, that was fascinating.
To cutting-edge gene therapies designed to rejuvenate telomeres, the possibilities for modulating these fundamental processes are expanding rapidly.
But as we've meticulously discussed throughout this deep dive, with each astonishing innovation comes a profound responsibility.
Absolutely.
It demands we carefully balance the potential for extended health span against the very real risks of unintended side effects, especially promoting cancer.
The ultimate goal, the guiding principle, is to extend health span without inadvertently accelerating the very processes like cancer that our genomic instability safeguards have evolved to hold at bay.
Right.
This demands a nuanced, cautious approach, learning from nature's evolutionary choices, studying real-world examples, observing effects in models, all while applying the most rigorous scientific and ethical frameworks.
So for you, as we wrap up this truly comprehensive deep dive, consider this provocative thought for your own reflection. As scientific progress allows us to increasingly understand and even intervene in these fundamental processes of genomic instability and telomere dynamics, how will our evolving definition of healthy aging shape the profound ethical and societal considerations for such powerful future interventions?
A critical question.
What new responsibilities, both individual and collective, will come with the astonishing ability to precisely repair and rejuvenate our very own genomes, not just for treating disease, but potentially for enhancing longevity itself?